comes science. They might be giants, here comes science.
Hey, Dr. H here. This video will be my Unit 7 review, which is all about evolution and natural selection. Okay, starting in part 1, I will give a brief overview of some of the big ideas of evolution, and then we'll get into the actual mechanism of evolution, both micro and macroevolution in part 2. Part 3, we'll go over the evolutionary relationships between all living things, that is the study of phylogeny. And then finally in part 4, something that I'm just calling broad patterns of evolution, we'll kind of take a look at some long-term evolutionary patterns that have existed since life really first appeared on Earth. Okay, so let's get started with part 1 and our basic overview. Okay, so certainly when we talk about evolution by natural selection, there is one name that we certainly cannot get away from, and that is, of course, Charles Darwin. Right? He is the one who is most widely credited with coming up with the mechanism for evolution by natural selection. And of course, he came up with many of these ideas, made many of his observations while he was traveling around the world on the HMS Beagle. There you see the ship. Coming off the ship, going on land, recording many observations. And some of the most important observations that he made were there off the west coast of South America in the Galapagos Islands. And some of the animals that, really caught his attention were the finches, right? All of these birds. What he noticed about these birds was every island had their own distinct beak shape. And this beak shape was adapted to the food source. Long, sharp beaks were good for eating cactus, right? Larger, stronger beaks were good for cracking seeds, and the short, pointy beaks were better for catching insects. So he sort of saw this interplay between the environment and the traits that the animals expressed. And he came up with this idea of natural selection, that the environment is really determining these favorable traits, and they tend to be passed on from one generation to the next. And one very kind of famous drawing or note in his notebook is this here, with the words "I think" above it. And you see, one of his first ideas of this, branching evolution that we are certainly very used to seeing now. We think about Darwin's idea of natural selection or evolution by natural selection. We can break it down, and really, it really is fairly straightforward, right? When we look at the combination of his own observations and some of the inferences that he made. A lot of them were based on previous work, right? And there's a large body of existing work from some of Darwin's contemporaries that had a very big influence on Darwin as he went around the world on his trip. But we'll just really focus on Darwin, right? And if we break it down and try to look at natural selection in its simplest state, there's this set of 5 inferences and observations, right, that we can sort of go through, right? So number 1, and this is just an observation that Darwin and, of course, others have made, that there is great variation among individuals, right? Even within a single species here, no 2 individuals are exactly alike. Number 2, every population has the ability to overproduce offspring, right? They can exponentially grow. However, they don't, because the resources are limited. So individuals have to compete for whatever resources are most limiting in that environment, right? Whether that's food, water, sunlight, there is this intense competition for the limited resources. And the winners of this competition, right, these successful individuals, will be able to reproduce and pass on those favorable traits, those traits that allow them to gather more resources within their environment. This has been going on for millions, or even in some cases billions of years of evolution. So there has been plenty of time for these small little changes to accumulate into large differences in the organisms. And that gives rise to this great diversity of life that we see currently on the Earth. So you oftentimes hear Darwin's ideas and natural selection referred to as survival of the fittest. But what I think a little bit better way to state that is not just survival, right? Survival is not enough. You need to actually reproduce, right? The most fit individuals from an evolutionary standpoint are the ones that are going to leave the most offspring, right? They are passing those genes on to the most individuals in the next generation, right? So really, we should be calling natural selection the reproduction of the fittest rather than just survival of the fittest. So let's take a look at some real quick examples of natural selection or of evolution. One is an example of natural selection, and then I have another example here of artificial selection, right? Something that Darwin did reference in his works. So for natural selection, we're looking here at this soapberry bug that is native to the southern U.S., right? I think it's down in Florida. And what has happened, a new food source has moved in, an invasive species has moved in, and the soapberry bugs are now using that as a new food source. So what effect has that had? Well, we see from the graphs there that the beak lengths have gotten shorter because this new food source, they don't need the longer beaks to get in and get to the fruit, right? So that environment has changed. So now the most adaptive trait has changed, and we see the phenotypes changing, right? There is a gradual move shorter. Okay, and then for artificial selection, artificial selection works much the same way as natural selection, but in this case, instead of the environment determining the most favorable traits, it is humans, right? We are determining what phenotypes, what traits we want in the offspring and selecting those and selectively breeding them. So here, these 5 crops all came from the same ancestral wild mustard plant, right? And the reason that they are also different is through artificial selection, right? They were bred, they were artificially selected by early farmers to show these certain traits, but they all came from the same ancestral species. So the last thing I want to talk about here with our introduction is just real quick going over some of the areas of evidence, right? How do we know that evolution has happened, right? It takes a very long time. It's a very slow process. It's very hard to actually see it happening in a laboratory setting. So what sort of evidence do we have that these evolutionary changes, that natural selection has actually gone on in the way that that Darwin proposed? So the one thing, the main thing that we look at, tying all these pieces of evidence together is similarities between organisms, right? There are striking similarities between many, many organisms that are closely related, and we can even see similarities in organisms that are not so closely related. So first, we have some anatomical similarities, right? Here we have the forelimbs of 4 different mammal species, right? And the bone structure is pretty much identical, right? Some sizes are a little bit different, but the overall pattern of the bones is exactly the same, right? So we would call these homologous structures, right? Because this shows that there is a recent common ancestor of all of these animals, right? Certainly these all are mammals. So once mammals started to diverge, that was fairly recent, right? So we call that evidence of divergent evolution, right? One common ancestor diverging out into multiple types of offspring. Okay, there's another type of anatomical similarity that we may see, and that is called analogous structures, right? And this is when organisms that are not really closely related to each other still show very, very similar anatomical features, right? And mainly this is because they are in very similar environments, facing the same evolutionary pressures. So they come up with the same kind of solution to their competition, right? To their evolutionary struggle. And this is why we call it convergent evolution, right? So we see an example here, the sugar glider marsupial from Australia, the flying squirrel, a placental mammal from North America, not very closely related, do not share a recent common ancestor, right? Even though they are both mammals, but anatomically they look very, very similar. Looking at another example here of with organisms that are a little bit further away, right? The fin structure of the shark, the wing of a bird, and the flipper of a dolphin, right? Very similar structures because they are all evolved to do the same thing, right? Swim as fast as possible, as efficiently as possible, right? But fish, birds, and mammals all have very different evolutionary histories, right? So this analogous structures here are an example of convergent evolution, right? Distantly related organisms kind of coming together at very similar structures. Okay, so that's anatomical evidence. Sort of related to that, embryonic relatedness. As organisms, especially vertebrates here, go through different embryonic development stages, they will look very, very similar, right? And the longer they look similar, the more closely related we think those organisms are, right? Certainly we see here the chick and the human at these very early stages share some very striking traits, and they're not that closely related. So after a few more stages, they're going to start to diverge more in their appearance. Our next area of evidence, and this is a very important one, the fossil record. Right, the fossil record is nearly complete. Right, we have really good fossils of many transitional species, and we can paint almost a complete history of many evolutionary lines. Right, it's not fully complete, mainly because fossilization is a very rare event, but we do have a relatively complete fossil record looking at many of these evolutionary lines. And our final piece of evidence, and probably the one that is the most important now, the most widely used now certainly, is our molecular data. right? Looking at DNA, RNA, or protein sequences and comparing the similarities between them, right? With the idea that when two species diverge, they begin to accumulate mutations independently, and the longer those two species have been apart, the longer they have been accumulating mutations on their own, and the less similar they would be. Right, so looking at the list of organisms here, right, humans and rhesus monkeys, right, both primates, very, very similar protein sequences, right, 95% similar between rhesus and humans, right. Going further away, right, looking at a frog, related, right, it's an animal, it's a vertebrate, but only 54%, because humans and amphibians, mammals and amphibians, have been separated for millions of years of evolution, so millions of years to accumulate all of those changes in the protein sequence. Okay, so that is our quick overview of the basics of evolution, and in part 2, I will get into the actual mechanism of how evolution works. Here I'm going to get into some of the actual mechanisms of evolutionary change, right? What are some of the processes that can actually cause populations to change over time, right? And we'll talk about two broad groups here. First, we'll talk about changes on a molecular level, right, on a genetic level, and that is microevolution. And then we will extend that and look at changes on a species level, right, and that is macroevolution. Okay, so we'll start here with microevolution, and this is changes in the allele frequencies, right? Changes in the genetics of a population, right? And there are a few forces that can cause that. I have them all listed here. And the reason I have a hand behind this picture here, because there is a great video, I will link it in the description below, called The 5 Fingers of Evolution. And it links each one of these processes to a different finger on your hand. A really super easy way of remembering all of these 5 processes, right? Because you can just look at your hand and remember what that process is, and kind of help you to recall all of these processes. So I'll go through these processes, right? And I kind of have the pictures there of the individual fingers. Please go watch that video, The 5 Fingers of Evolution. It really helps make things make sense. All right, so the first one here is genetic drift, and this is changes due to chance, right? Randomness can have a large effect on populations. And generally what we see is smaller populations are much more prone to these random events, right? These random changes in gene frequency. So, for example, here is a simulated data set for a 50 generations of a population, just looking at random allele frequency changes. And with a very small population of only 20 individuals, we see that there is, random chance, again, has a very large effect. Increasing that population up to 200 individuals, the allele frequencies do not drift quite as much. And increasing it again up to 200 individuals, we see that over these 50 generations, the allele frequencies stay fairly constant, right? There is a little bit of change, but overall the diversity is nowhere near as great as we saw with the smaller populations. So the other piece that we need to think about with this genetic drift and the small populations is what would cause a population to become small, right? And there are a few events that could happen. One is what we call the bottleneck effect, and this is when an initially large population goes through some drastic event where the population now becomes very, very small, right? And not only is that small population now much more likely to be affected by random chance, the allele frequencies in that surviving population may not accurately represent what the initial starting population was. Okay, so that is the bottleneck effect, right? A large population becoming very small. Kind of related to that, and these two are often confused, the founder effect. Okay, and this is when a small portion of a population breaks away and moves into a new area, and no longer exchanges genetic information, no longer reproduces with the original population. Right, that new population there, right, those few individuals that left will probably have a slightly different gene pool than the initial larger population. Okay, so this is, the founder effect is when a portion of a population leaves and goes to a new area and sets up a new population. The next factor that can cause allele frequencies to change, non-random mating, okay, or what we refer to as sexual selection. Okay, and this is when mate choice is determined by factors that may not actually affect survival, right? But it's on some other criteria. And we really see this a lot in birds. They're always the example that we give a lot. And we certainly, when we think about the peacocks there on the left versus the peahens, right? The peacocks are bright colors. They have the big, flamboyant, showy tail feathers. Peahens don't have those, right? And this is a result of sexual selection, and it is called sexual dimorphism, right? And basically that just means that the males and the females of a species will look very different, right? Male peacocks, female peahens, right? And sexual selection can be very, very powerful, right? This mate choice can lead to all kinds of very interesting evolutionary outcomes. All right. The third factor that can cause microevolution, mutations, right? Changes in the DNA sequence, right? And that's very straightforward. If there is a change in the DNA sequence, right, and that leads to a new allele, that new allele is now going to affect the ratios, right? And mutations can happen all along a gene. Right? Not all of them are going to have effects, but mutations are happening all the time. All right, our fourth factor here, and this is our final one that is actually random, is gene flow. Okay, and gene flow is the movement of alleles into or out of a population. Right? As adjacent populations exchange individuals, and they reproduce with each other, they're going to be exchanging genetic information, right? That tends to lessen the differences between the populations and make the populations more similar, right? And a quick example of that here, an illustration of that, these two snake populations on the Ohio mainland and on the Ontario mainland, and then on an island very close to Ohio, right? The snakes can travel between the Ohio mainland and the islands, so they are reproducing. The Ontario mainland, a little bit further away, that population is more isolated. So on the Ohio mainland, we see these forms, right, B, C, and D, the banding patterns that are not present on the Ontario mainland, but are present on the islands, right? Because these genes are constantly being moved back and forth, right? They are these two populations here are reproducing with each other. Okay, so those four, right? Those first four that represent the first four fingers. None of those are adaptive. The only process that leads to adaptive change in a population is natural selection, right? And that is when a gene is adapted for survival in the environment and leads to better reproductive success, right? Leads to more reproductive fitness. Okay, and when we look at natural selection, there's a few different, three different basic patterns that we can identify. So if we imagine we start with our original population here of mice with a nice distribution of fur colors, as evolution proceeds, right? So you imagine these are moving into a new environment, so they are now going to evolve and adapt to that environment. That evolution, that natural selection, can be directional selection, where one of the extremes is selected against, so the population average kind of shifts in one or the other direction. There is disruptive selection, where the most common trait is selected against and the two extremes are selected for. So the two extremes would become more common in the population. And then finally, there is the stabilizing selection, where both extremes are equally selected against. So that average, right, the most common trait is then selected for and becomes much more common, right? And we kind of see the bell curve sort of tightening up there. Okay, so this natural selection, that is the only one that actually leads to evolution in response to the environment. One final thing, looking at microevolution and allele frequencies, something that may very well show up on the test, and that is Hardy-Weinberg equilibrium, right? How do we measure these gene frequencies and try to keep track of whether they are changing? Okay, and for Hardy-Weinberg equilibrium, there are 5 factors, 5 requirements that must be in place for a population to be in Hardy-Weinberg equilibrium. You see them listed here. They are the exact opposite of the 5 things that would cause a population to change, right? That would cause a gene pool to change. So watch the 5 Fingers of Evolution video, learn those 5 things. You will also learn these 5 conditions for Hardy-Weinberg equilibrium. Okay, there are 2 equations that may show up with Hardy-Weinberg. The biggest issue that I think students have with this is remembering which equation refers to individuals and which equation refers to alleles. And now they define P and Q for you here, right? They say P is the frequency of the first allele, generally the dominant allele in a population, and Q is the frequency of the other allele, right? The recessive allele in the population, right? So that should tell you that the equation P plus Q equals 1 is what we should use if the problem is asking about allele frequencies. If instead the problem is asking about individuals, right, and how many homozygous dominant, how many heterozygous, how many homozygous recessive, then we would use the other equation, right, the squared equation, right, P squared plus 2PQ plus Q squared equals 1, right? And in that equation, P squared is the frequency of homozygous dominant individuals, 2PQ was the frequency of heterozygous individuals, and Q squared is the homozygous recessive individuals. The other important thing to remember, and something that will often lead to mistakes, is in a case of complete dominance, right? Remember that the homozygous dominant and heterozygous individuals are going to look exactly the same, Right? So if you are given the frequency of the dominant trait, that's not going to be, you can't really do much with that in terms of Hardy-Weinberg. You actually have to start with the frequency of the recessive trait and find Q squared. Make sure to use the equation sheet, right? They give it to you. You don't need to memorize these things. And the information to remember which formula is for alleles and which is for individuals is actually given to you right there, right? Because it tells you that P is the frequency of the dominant allele, and Q is the frequency of the recessive allele. So that's microevolution, right? Changes on a genetic level, right? Changes in allele frequency. So the other piece now is macroevolution, right? The formation of new species, right? And this occurs when microevolution has occurred, and we add in this reproductive isolation, where the two populations that are diverging now no longer reproduce and no longer exchange genetic information, right? So there's no more gene flow, right? And that is going to allow these species to diverge and become new species. Before we talk about formation of species, I want to take a minute and actually define species, right? Because it can be a little tough, right? The two birds there look very similar, but they are different species, and all of the humans there on the left all look very different, but they are the same species. What is our definition of species that we could use that would allow the two birds to be kept separate, but all of those people to be put together as a species? So the definition that we use is called the biological species concept, right? Where individuals are considered the same species if they can successfully mate, interbreed, and produce fertile offspring. There are barriers, right? Reproductive isolating barriers that will keep species apart. And these barriers are classified as to whether or not they are prezygotic or post-zygotic. Right? Remember, the zygote is that initial fertilized egg, right? When the sperm and the egg fuse during reproduction. So prezygotic barriers would happen, either they would block a mating attempt, or if a mating attempt does occur, it prevents successful fertilization. Right? The post-zygotic barriers are after a fertilization event has occurred, but the offspring then do not go on and make a new and represent a new species. Okay, so we'll talk first here about some of the prezygotic barriers. Right, so these are going to either prevent a mating attempt from occurring, or if a mating attempt does occur, they are going to prevent the sperm and the egg from successfully fusing and fertilizing. The first set here are the ones that will actually block the mating attempt. Right, so these are things like habitat isolation. Right? The species just live in different areas of the world. Temporal isolation. They reproduce at different times of the year. Or behavioral isolation, right? There are specific mating rituals that animals perform, and different species will not perform those same rituals, so that will prevent a mating attempt. Okay, other prezygotic barriers occur after a mating attempt has occurred, and these will prevent fertilization, right? So the sperm and the egg will not successfully fertilize, even if there was a mating attempt. So mechanical isolation there, the reproductive structures have to line up, right? If they're not compatible, the sperm and the egg are not going to fuse. And then gametic isolation, this is especially important for species that fertilize externally, where in aquatic animals or aquatic species, where the sperm and the egg are released out into the water, there is a receptor and a ligand, the receptor on the egg cell and the ligand on the sperm cell, and they have to match up, right? And they will only match if they are the same species, right? So different species will have different ligands, different receptors, and fertilization will not occur. Okay, so those are the pre-zygotic barriers. The post-zygotic barriers, right? So these occur, right, after a mating attempt, after a successful fertilization, and oftentimes we'll see that it's the reproduction is successful in terms of creating an offspring, but that offspring then cannot go on and survive or cannot go on and reproduce. So, like I said, these are after a zygote has formed. So we have three big ones here. Reduced hybrid viability. So this is when the hybrid individuals are just not able to survive well. Reduced hybrid fertility. That means that the offspring are sterile, right, and cannot reproduce themselves. Remember that the biological species concept says that the offspring have to be fertile. And then hybrid breakdown. That one's a little different. That one is the first generation hybrid species looks fine, looks very healthy, but when those hybrids try to reproduce with each other, the offspring, that second generation offspring, are not healthy. Okay, so those are the main post-zygotic barriers that maintain this reproductive isolation that is very important in keeping species separate, right? When in microevolution, we talked about gene flow, right, and how gene flow works to make populations more similar, right? To have divergent evolution, there has to be this reproductive isolation, right? And it's some combination of multiple prezygotic and post-zygotic barriers. Okay, and the last thing I want to talk about here, just some real quick patterns of speciation. Right? What is the one of the main things I'm going to talk about here? What is the geographical relationship between the two populations that are diverging? Right, and we call this either allopatric or sympatric speciation. Right, in allopatric speciation, there is some geographical barrier between the two species, right? That is going to prevent them from mating, right? Prevent gene flow between these two populations. So in this case, we see that the pond here has evaporated, like the water level has gone down, and now there are two separate ponds, and the two populations of fish are now separated. Right? That is allopatric speciation. In sympatric speciation, there is no geographical isolation, so there is some other isolating mechanism that comes into play, right? So these two species here that are diverging, these two populations, share a geographical range, right? But one of those other reproductive isolating mechanisms, right, one of those other barriers has come into play here and are preventing these two populations from exchanging genetic material. And the final thing that I want to talk about here is a couple ideas about the rate of evolution, and two different thoughts, two different kind of schools of thought about how evolution has worked long term. They are the punctuated model and the gradual model. These are two just ways of thinking about, well, how has evolution happened? Right? So in the punctuated model, that says that there are these rapid bursts of evolution where many species are formed around the same time, and then they are very stable for long periods of time. Right? In the gradual model, that says that change is very gradual, that there is a constant low level of change, and species are slowly changing and getting into their full divergent final forms through this constant slow rate of change. Right? Which one is true? Again, very hard to say. Evolution can be very tricky to study because it is generally a very slow process. My thought is that the reality is probably a mixture of these two models. that is our mechanisms of evolution. In part 3, the next part, I will talk about the end result of all of this evolution, and that is the great diversity of life and the study of that diversity, which is phylogeny. Now I am going to talk about sort of the end result of all of these billions of years of these evolutionary processes, and that is basically the great diversity of life that we see around the globe. Right? And we see here are kind of a basic representation of our tree of life, right? How are all living cellular-based life forms on Earth related to each other? And we have the 3 major domains, right? Domain bacteria, domain archaea, and domain eukarya, or the eukaryotes. And about 3.8 billion years ago, there is that last universal common ancestor. I'm not going to get into all of these pathways here, right? That we could explore this tree really forever and find all kinds of interesting things to investigate here. What I really want to focus on is how these trees are built, right? What sort of data, what is the process behind building these relationships? And this, the science of discovering and investigating these evolutionary relationships between organisms is called phylogenetics. Finding these evolutionary pathways that kind of lead back in time to find how two current species are related. What is their common ancestor? And when we start building these relationships, one thing that we want to try to develop is a clade, right? This group of related species, and not only putting the species together, but also uncovering how they are related to each other. And there's a few different ways that these relationships can be represented. One here is called a cladogram, or I guess a cladogram. And this basically shows a relationship here with a few characteristics along the bottom. And as they go, right, any organism that is past that trait is going to display that. So, for example, if we look at lungs, right, lampreys and sharks do not have lungs. Everything beyond that would, right? So salamanders, lizards, tigers, gorillas, they all possess lungs. Right? Now, a common misconception here is to think, well, okay, this means that a tiger, evolved into a gorilla, right? Which is certainly not the case, right? That wouldn't make any sense whatsoever. And that is not what a cladogram would tell us, right? Something that we could take from this cladogram, we could say that tigers and gorillas are more closely related than a salamander and a gorilla, right? Which would certainly make sense, since tigers and gorillas, those are both mammals, right? A salamander is an amphibian, so it's a little bit further away. The way that these cladograms are built, we need to create a little data table here, or a character table as it's called here. And we would basically score each organism as to whether or not it shows each character. Right? And here, on the left-hand side, there we see the characters that were chosen for this particular set of organisms, and everything is scored with either a 0 or a 1, and the scores are added up for each organism, and then they are ranked and placed on the cladogram. Over on the right-hand side, we see a slightly different way of representing the same data, right? And this is on a phylogenetic tree. And the phylogenetic tree attempts to show a little bit better representation of the evolutionary changes, of the evolutionary relationships between our group of organisms. Right? So in this case, we can trace back and say, okay, a turtle and a leopard have a more recent common ancestor than a leopard and a bass. A very important consideration when we're putting organisms together in groups is that the group, or the clade, is evolutionarily valid, right? And an evolutionarily valid clade must be monophyletic, right? And that means that it contains the ancestor, right, and everything that descended from it. So let's look at some examples here of a valid grouping and some groupings that don't really hold evolutionary value. Right, so our yellow grouping here, that is our monophyletic group. This is an evolutionarily valid clade, right? And this would include, this is the reptile clade, right? So you see it includes the turtles and tortoises, the lizards, crocodiles, and birds. The blue shaded clade there would include all those reptiles except for birds, right? Leaving birds out. And you can see, right, when we draw it visually like this, that grouping really doesn't make any sense, right? And an even worse grouping is that red one there, grouping all of the four-chambered heart, warm-blooded animals together, right? So the mammals and the birds, right? Those are very far apart evolutionarily, but they are often grouped together because they do share a few characteristics, right? Warm-blooded, four-chambered heart. But evolutionarily, they are not that closely related. So I don't think it's terribly important that you understand the exact definition of the two incorrect groupings there, right? The paraphyletic group and the polyphyletic group. It's more important that you understand the definition of the correct grouping, right? The monophyletic means that it has the common ancestor and everything that evolved from it, right? So the ancestor and all the descendants. That is the evolutionarily valid clade, right? The monophyletic clade. The way that these phylogenetic trees are built today, right? The modern phylogenetic trees are mainly built using molecular sequence data, right? Either DNA, RNA, or protein. Right? So the way this is done and the idea behind it, right? At the beginning, before the species diverge, there is one sequence, right? They are a single species, and they all will share their sequence. In this case, we are looking at DNA sequence. As the species begin to diverge, as the reproductive isolation takes place, the two populations, right, the two sequences here, will begin to accumulate individual mutations, right, independent of each other. So we see in population 1 there is a deletion. In population 2, there is an insertion event, right? So, and these are happening independently. So now, when we just look at these sequences, they don't really look too much alike anymore, right? But these can be plugged into computer programs that have been written to align these sequences that maybe just by looking by eye don't have that much in common, but the programs can find these similarities and can then score them and figure out, assuming that there is a stable mutation rate, right? Like we talked about back with evidence of evolution, the longer the two populations have been apart, the more mutations, the more differences they will have accumulated. So by throwing a bunch of the sequences together, the program can then calculate all of the evolutionary relationships and create the phylogenetic tree. One aspect of building these trees that we need to take into account is this idea of maximum parsimony or parsimony. I'm not really sure how it's pronounced. I've heard it both ways. And this is basically the idea that the simplest explanation is usually the correct explanation. Right? So when we are putting trees together and figuring out mutation rates and how many changes have accumulated, we want to make sure that we have the fewest independent evolutionary events. Right? So here's an example of that. Looking at 3 representative organisms from the 3 major eukaryotic kingdoms, right? Animal, plant, and fungus. The top there shows percent differences in a gene sequence. So which of the 2 trees there at the bottom is more likely? Well, the tree on the left there, the one that's labeled more likely, the mutation rate is fairly constant throughout that, right? There's the first divergence, and the one side has a 5% change, and then there's another branch, and each of those branches then accumulates 15% mutations. So 15 plus 5, right? That equals 20%. And on the other side, right, on the other arm going up to the plant, we see that adds up, right? It's 20%. So everything adds up, and we have a stable mutation rate. Looking at the other tree, we have their first branch event. The fungus go off on one side, accumulate 5% mutations, and then stop. Right? Stop evolving. And then on the other side, we have another branch point, and animals go off, humans go off and divert and accumulate 15% mutation rate, and then they stop. And then the plants go up and accumulate another 25% mutations, and then they're done. Right? Too many things going on there. Right? Really complicated explanation of why all these mutation rates are different. Okay, so the simpler explanation is generally the correct explanation, unless we have lots of other data to support the alternate explanation. For example, looking at the evolution of the 4-chambered heart. Right, this is a complex anatomical feature that we know has evolved twice. Right, it evolved separately in birds and in mammals. Right, and putting birds and mammals together in one group, separate from the lizards, doesn't make sense. Even though looking at only that one trait, it does make the simpler tree. We know, due to an overwhelming amount of other data, that bottom tree is the correct relationship. That birds are part of the reptile group, and so are closer related to the lizards than they are to mammals. And the anatomical feature, the 4-chambered heart, that they have in common, did actually evolve separately twice. This is a look at the overall weight of all of the data, right? Not just one particular trait. Okay, one last thing here to kind of talk about is the fact that, the trees that we look at, like I showed at the beginning of this section, are greatly simplified. Right here, we see a little bit more accurate version of the 3 domains, showing all of this horizontal gene transfer, right? Genes moving from species to species, right? Eukaryotes, that really hasn't happened for a very long time, right? So really since the introduction of the chloroplast in the algae and plant groups, but certainly in the other domains, the archaea and bacteria domains, we see that there is lots of horizontal gene transfer going on there, and there probably is still a good bit going on now. Okay, so that is the end of our phylogenetics, right, and the evolutionary relationships between all of the different living things. In the final part of the video here, I'm going to take a look at evolution from a more kind of a long-term scale, something that I'm calling the broad patterns of evolution. So we'll look at a couple things here. The beginning of life, the origin of life, that is abiogenesis, and then just an overall history of life on Earth. Abiogenesis, that term literally means the beginnings of life from no life, right? So what was this first life form that appeared on Earth? Do we have any clues as to what it may have been like, what it may be similar to that is currently living now? And we don't have any direct evidence for these first living cells. They certainly didn't fossilize, but we do have some indirect evidence, right? Some remains that we hypothesize were probably due to the presence of living things. And we've dated these to somewhere between 3.5 and 3.8 billion years ago. We do have some good evidence, though, about conditions on this early Earth. And we know that conditions there were very different from the current conditions in terms of the atmosphere, in terms of the overall climate. Thinking about the atmosphere, the gases that were present were very, very different. Now we have what is called an oxidizing atmosphere, right, with a good bit of oxygen present. But 3.5 billion years ago, there was very little or no oxygen present. And you see all of the other gases that were available in the early atmosphere. So there were a set of experiments done, kind of looking at these early atmospheric conditions and what that meant in terms of production of early organic compounds. This is referred to as the Miller-Urey experiment for the two researchers that led this group. And they replicated these early environmental conditions with a certain set of gases, introduced energy in the form of a electrical spark, right, to replicate lightning strikes in the early Earth atmosphere, and just ran this through this closed system of glass equipment over and over, and would periodically take samples and determine what they found. And what they saw was that there were some fairly complex organic molecules that would actually spontaneously form in these conditions. They found amino acids, they found a few different nucleotides, I believe they even found some more complex lipids, kind of showing that it is possible for these basic building blocks of life to have arisen from a completely abiotic, right, non-living environment, one that is, of course, very different from the current Earth environment. One other very interesting piece of evidence about how early cells could have formed was taking some of these molecules from the Miller-Urey apparatus here and dropping them onto a particular type of hot clay, right? That volcanic clay that would have been present or believed to have been present on this early Earth. What they found was that under these conditions, they would spontaneously form these little tiny vesicles, right? That little bubble-shaped, membrane-bound, what look to be early cells, right? And these vesicles can actually bud and sort of do a very primitive version of reproduction. And they can also sort of absorb RNA molecules or RNA nucleotides that are in the surrounding environment. And we have a lot of evidence now that a lot of these early cells, all these early life forms, were actually RNA-based instead of our current DNA-based genetic material and proteins performing much of the biochemical reactions in a cell, these early kind of proto cells where everything was done with RNA. Okay, so we do have some good evidence here about the way at least some of these molecules may have started to form, and that under certain conditions they can spontaneously arrange themselves into structures that resemble living cells. So what about, so that's 3.5 billion years ago. So what about, from 3.5 billion years till now? What has the history of life really looked like? And obviously this history of life goes very far back. We do have good fossil evidence of many living things, right? Not all the way back to those first cells, but for much of the history of life, we do have a pretty good fossil record. We have broken up the geological timescale into different timescales, right? Eons, eras, periods, epochs, right? You can see the list there. The names of these and the dates are not terribly important. It's just kind of giving us some reference to kind of talk about all of these things. This timescale goes back so far, right? It's very hard for us to kind of conceptualize. And there are a few different ways that we can represent this long time scale. Right here, we see it laid out on a straight timeline. Right, you see some of the important events there. Right, oxygen being released into the atmosphere. Right, about 2.7 billion years ago. Right, that would have been when photosynthesis first evolved. Moving forward, looking at some of the more recent events. Right, single-celled eukaryotes, the Cambrian explosion, when many of the current animal forms, a lot of the animal phyla were first set up. And there at the bottom, you can see sort of trying to represent this time scale as a clock. Right, so if we say that the solid Earth formed an hour ago, right, when did all of these things happen in terms of time, right? And I think it says somewhere in there that humans were 0.2 seconds ago, right? So it kind of gives you an idea that Earth and life have been here for a very long time, long before humans were. Some of the events that occurred during the history of life that are very important, not only, as shown here, some of the appearance of new forms, but also the mass extinction events, right? And that the red line here on the graph shows the extinction rate. And we see marked out there the 5 major mass extinctions that have occurred through Earth's history, right? With the most recent one there being around 65 million years ago, between after the Cretaceous period, right? And that's the one that wiped out all of the large dinosaurs, right? The largest mass extinction was about 252 million years ago, and that is referred to as the Permian mass extinction, right? And actually marked the end of the Paleozoic era. And you see the extinction rate went way up, and the blue line there, the number of surviving families, you see how rapid a fall that takes. But one thing to keep in mind with these mass extinctions is for the organisms that survived, for these species or families that survived, these mass extinctions often will lead to a very rapid burst of evolution, because now there is whole new areas to go and explore and new resources to exploit, or the predators have disappeared. So now there's not this big threat to survival, and the prey species can expand their geographic ranges and go and, evolve. So if you look closely at that blue line, you'll see after every mass extinction, there is a fairly rapid jump up again in diversity. So while these mass extinctions obviously are very negative for many species, right? Those, especially those that go extinct, they will also have a great opportunity for the animals or the all of the organisms that survive, right? And not just the prey species, but also the predator species will take a large jump in evolution. Right, so looking at that Permian mass extinction, right, the 252 million years ago event, this graph is looking at the percentage of marine animals that were predators. And for a long time before the Permian mass extinction, that number was fairly stable, right? Somewhere around 15%. Immediately after that mass extinction, immediately after the Permian period, see that number jumps way up and then kind of stabilizes at a slightly higher number, right? Around 22%, right? Which is a fairly significant rise when you look at the overall graph. So that sort of indicates or gives us more evidence that these mass extinction events lead to a large burst of adaptive radiation as these surviving organisms, right? In this case, obviously these would be animals. The surviving animals, right? In this case, go out and explore new resources, right? Adapt to the new landscape. That is the end of our Unit 7 review. I hope that all made sense, and good luck on
Girl named Lizzie.
Dropping science like Galileo dropped the orange.